FTP. And then here it shows you have the, this would be the ovolysin. Then you have the PROTAC here, which this is the full PROTAC. And then you have the linker. And then you have the IPP peptide, which will, oh, sorry, no, I did it backwards. This is the IPP peptide, and this is the ovolysin. So then you have here showing the ubiquitin tagging the target protein, and it's going through the process of ubiquitination into the proteasome and coming out as smaller peptides. So then here you have the PROTAC synthesis. And I guess the important part that I want to highlight is this part right here, this ketone. So the NABH4 will, it reduces this ketone so that a linker can be added to this section. Like this is the final linker, but this is the beginning of the linker. And I think it was purposeful that the linker is kind of facing away from the structure itself and kind of outwards so that it's easier for the linker to attach to the E3 ligase. And you can kind of see that better in these pictures from Chimera. So right now what you're seeing is this white part. The white part is the MET-AP2, and the rest of it's the ovolysin. So you can see that maybe it's better in this picture, that you can see that the linker for the ovolysin is kind of facing towards some sort of space so that it's easier for the E3 ligase to attach, and it kind of prevents and ensures any problems with attaching to the E3 ligase because it's such an important step.
Mm. You can see the covalent bond there too.
Mm-hmm. Yeah.
That? Yeah. That probably worked to their advantage quite a bit, actually.
Mm-hmm. So the paper was kind of broken up into 3 sections, kind of describing the process of how they got the PROTAC to work and what they did. So I'm going to talk about the first 2 sections of this process. So initially what they were looking at was, does MET-AP2 bind to the PROTAC? So they used a Western blot analysis test to kind of get this data. So what you can see here is that at different concentrations of the PROTAC, the MET-AP2 binded to the PROTAC or it didn't bind to the PROTAC. But the point is that at some point it binds to the PROTAC so that it does work. So here at 10 micromolar, the MET-AP2 does bind to the PROTAC. And then at 10 millimolar, the MET-AP2 does bind to the PROTAC. And these last 2 here, where you have like the plus with the IPP and the plus with the ovolysin, it's just showing that the one component of the PROTAC was added. So this is the full PROTAC here, this blue line and then the yellow. But what they're showing here is that only the IPP was added to the MET-AP2 and it didn't bind. The MET-AP2 didn't bind to just the IPP, and the MET-AP2 didn't bind to just an addition of ovolysin. So then in figure B.
So can I jump in for a second, Naya? So,
yeah.
So your friends here might not be accustomed to looking at protein gels. So okay. Does anybody have any questions about like what the evidence here is for the PROTAC binding to the MET-AP2 protein, the target protein?
Why are there 2 bands for some of them?
So, okay, so the 2 bands, the band at the bottom is just indicating that there's MET-AP2 in all of these, added to all of these concentrations of PROTAC. So then this, the other striations are because of that at this point, the MET-AP2 does bind to the PROTAC, which is what you can see over here. And I guess the differences in the shifting of the bands is because the molecular weight of the PROTAC is changing. So it's creating a darker striation. And the IPP peptide is already kind of big, which is on the PROTAC. So it's creating more shifts.
Yeah, that's exactly right. Like in this case, the IPP, you're adding mass to the protein, and that's reflected by, you can see that by a difference in its migration on the gel. Yeah.
Mm-hmm. Okay. So then in figure
So
B,
I'm sorry for keep interrupting you, Naya,
You're
but
fine.
that might not happen in the future because I think, not to jump too far ahead, but people don't use peptides as E3 recruiters or E3 binders anymore, just because so the evidence that people will use for binding will look different in the future rather than this shift in the gel because of the molecular weight. You know what I mean? Yeah, because peptides are not useful as they're not usually cell permeable. You certainly can't turn like a peptide into a drug because your body would just like sort of digest it, really. So even though this is like the first one, but I think everybody knew that to move forward, they have to get away from a big peptide as the E3 binder. So,
so then in figure B, it's essentially the exact same experiment. Like you can see here that at the second blot for B is the exact same results as the fourth blot for A. However, what they did was in the third blot, they added the PROTAC, the full PROTAC, but then they also added an addition of IPP. So what you can see here by this dark striation is that the MET-AP2 was still able to bind to the PROTAC, even though an addition of the IPP peptide was added. And then in this next blot over here, you can see that there was an addition of ovallicin. So along with the PROTAC, they also added ovallicin. And you can see that since it's lighter, that the MET-AP2 either didn't bind to the PROTAC or didn't bind as well to the PROTAC when ovallicin was added. So what they kind of concluded from this was that an addition of free ovallicin interfered with the binding of MET-AP2 to the PROTAC.
Yeah.
And then the next step they did was they wanted to see if the PROTAC can recruit the MET-AP2 to the SCF. So essentially what they did, SCF also stands for E3 ligase. So essentially what they did was they took extracts from T93 cells that were either for a control group or plasmids that contained this flag epitope. And that flag was tagged with CUL1 or beta TRCP. So like how I mentioned before, the CUL1 and the beta TRCP were components of the E3 ligase. So essentially it's coded with E3 ligase. So then these cells, they were immunoprecipitated with anti-flag resin to get these small beads. And so these beads were either control beads or they were coated with this E3 ligase. So then after that, the beads were then placed in a mixture of MET-AP2 that was attached to the PROTAC. And they wanted to see if the MET-AP2 would bind to the beads that were coated with the E3 ligase. And what you can see here from this bound area is that the MET-AP2 did bind to the beads that had the SCF and with the SCF beta TRCP coated on them, so that it does work. So that the right now as of this step, the PROTAC is able to kind of guide the target protein to the E3 ligase to begin the process of ubiquitination or proteolysis. And that's all.
Yeah, you can clap. Very nice. I have a question, Naya.
Yeah.
Can you go to your final slide, please? Yes, that one. Now, I'd like to sort of imagine that, okay, so can you think of an experiment that they could do here that's analogous to the experiment that they did on the previous figure, that would, and then predict maybe what its result would be? So you remember how in the last time they added free ovolysin and free IPP peptide, and they saw that the ovolysin, exactly like you said, it interfered with the PROTAC binding to MET-AP2. What could they do here maybe that would be an analogous experiment, and then what would you predict the result would be?
Well, I would probably think if they added additional IPP that the MET-AP2 would still be able to go towards the SCF, but that if ovolysin was added, that probably wouldn't be the same case, that there would probably be some sort of blockage for the PROTAC being able to recruit the MET-AP2 to the SCF. And I don't think that they would probably see like a lighter bound area here.
For which one?
Wait, what do you mean which one? For the
ovolysin or the IPP peptide, or both?
So you would probably see a darker line for the IPP, and then you would probably see a lighter line for the ovolysin.
I disagree.
Really? Why?
I think so. Well, so think about, so when they did it in the first experiment, they were adding sort of each half of the PROTAC as like a competitor. And in that first experiment, which I think was figure 2, the binding interaction they were measuring was between the PROTAC and MET-AP2, which is mediated by which half of the PROTAC?
Well, that's ovolysin.
Correct. And ovolysin blocked that interaction. Free ovolysin, you know what I mean? So what's the key interaction that they're probing for in this experiment? Because they have the E3 ligase trapped on the bead, right? And what they're adding to it, I think, is actually the preformed PROTAC MET-AP2 the bound complex. Like I think they already pre-incubated that and then added it. Do you see what I'm saying?
That's when those like the beads that they got from the from putting in all the anti-flag resin, they added that to a mixture of the MET-AP2 and the PROTAC.
Correct.
They were already like, I guess, connected.
Yeah. So what's the key interaction then that would bind MET-AP2 to the E3 ligase coated beads in that case?
I guess the key interaction would be if the ovolysin can attach to the MET-AP2.
I think they already did that.
Oh, so, okay. So you're saying,
like I think that the PROTAC was, they pre-attached it to the MET-AP2.
Yeah. So, I guess I think I'm kind of confused what you mean by like the key interaction. If it's already like there, and if like the whole point of it's to recruit it to the MET-AP2, like recruit the MET-AP2 to the SCF, wouldn't that just be the key interaction? Like that's what they're looking at here.
But what's the component though that binds to the SCF?
Oh my goodness, it's the IPP peptide.
Yeah, you follow me. So like I think what they started with was MET-AP2 that basically already had the PROTAC on there. So it's like dangling off of it is the IPP peptide. Right? It's like actually it's attached because it's on the PROTAC to MET-AP2. And then they wanted to see if that free protein, the MET-AP2 that's bound to the PROTAC, was able to stick onto these E3 ligase beads or not. Yeah. So I think what they didn't do it, but they probably should have, right? Done the same thing where if they added each free component, they could have made this a little bit more of a convincing figure, I think. Yeah. Like they did in the last one. Does anybody have any questions? for Naya? Do you all get the idea so far of what PROTACs are designed to do? Yep. Yep. Everybody's nodding.
Like slowly figuring it out.
Yeah, I think, like I said, Naya had one of the hardest sort of lifts in the beginning. But then after everybody sort of sees how it works in general, all the modifications after that are kind of minor and just are these smaller improvements, not unimportant improvements, but they're all the subsequent molecules. They all do the same thing, just like maybe more efficiently, or the molecules are more drug-like or stuff like that. Yeah. Great. Thanks, Naya.
No problem.